disease, identifying a drug or other type of therapeutic that will bind to this protein and change its function in some way. And since this is such a core part of drug discovery, we really think that deep mutational scanning types of technologies can really have a huge impact in the area of drug discovery and development. In particular, there's a number of questions that come up in drug discovery efforts that DMS could potentially provide really nice answers to. The first is, it's safe to actually change this protein's function in people. The second is, which protein functions are actually implicated in disease, and therefore which ones do you actually need to which functions do you actually need to target with a drug? The third is, which parts of the protein are important for its function or for drug binding? And then the fourth is, are patient mutations in the protein likely to affect whether or not a drug works? And we had a nice explanation of this morning in the context of drug resistance for chemotherapeutics. We are actually looking at this from a slightly different angle, which I'll talk about in more detail throughout the talk. A lot of the drugs that we're developing are chaperone therapies to correct misfolding variants in proteins. And so not every patient mutation that causes a misfolding variant is actually going to respond to one given chaperone therapy. And so really understanding which variants are going to respond can help you identify which patients are going to be most likely to benefit from your therapeutic. And then finally, is there any other additional information about the protein, such as allosteric sites, that could help us identify or design better drugs faster? I'm going to, for the purposes of this talk, give you some examples from one particular drug program that we've used deep mutational scanning in. And to give you some idea, like at a high level, kind of how we're thinking about incorporating this into discovery programs. And this program was to try and identify drugs that could target MC4R, the melanocortin-4 receptor. This protein, which is shown up here in green, is a GPCR, or G-protein coupled receptor. It's localized in the membrane of the cell. It's activated by a small peptide called alpha MSH. And MC4R, when activated by this peptide, responds through a couple of different GPCR signaling pathways. For the purposes of this talk, I'm going to focus on two of them, which are termed the GS signaling pathway and the GQ signaling pathway. Rare loss of function variants in MC4R are associated with obesity and collectively actually form the most common monogenic form of obesity in humans. And therefore, targeting this particular protein has been an interest for a lot of drug discovery efforts. All right. So in order to use DMS for drug discovery, as I'll talk through some of the applications later on, we need really sensitive and robust assays. And so I want to talk through kind of like what we're doing to develop really robust and sensitive assays. So the first thing that we need is a robust assay that assesses disease-relevant mechanisms. And so to do this, we are using DNA synthesis to make every possible single amino acid substitution linked to a barcode gene, or a barcoded reporter. So we make a library of all of our variants of our protein. They're each linked to a unique barcode that is in the 3' UTR of a reporter gene that's under the control of a response element that's going to respond to some functional effect of the protein of interest. And so the reason we use this is because we can actually directly assess cellular signaling functions that are implicated in disease. And so this is the example from our GS assay where we've taken each of our variant barcode libraries, we've integrated one construct per cell in HEC293 T cells. If the variant that's produced is functional, the MC4R will be localized to the membrane. It'll respond to alpha MSH. This will then activate the GS signaling pathway, which will activate CREB and bind to the CREB response element in our reporter. We can then use sequencing of the barcodes in the reporter transcript to recapitulate the map of the effect of all of the different functional variants of MC4R. We, in most cases, can use response elements to different signaling pathways kind of straight out of the box. But in some cases, those response elements don't have really robust signal. And so we really lean into a lot of synthetic biology tools, such as developing relay systems, one of which is shown here, to improve the assay signal to noise if necessary. And so this relay system is based on a GAL4 VPR fusion, which amplifies the signal of this particular reporter. So this is something that we had to use to actually develop a good GQ assay. So we were able to develop two assays for this particular protein. Now we need to know that we have the power to detect subtle effects. And so we put a lot of thought into modeling the power, or the number of barcodes that we would need in order to detect variant effects of different sizes under different potential conditions. And the short answer is that we really target at least 30 barcodes per variant to have good statistical power for detection. And this should be good to identify even partial loss of function variants. For both of our assays, we did a pretty good job with this. For the GQ assay, we were able to get 28 barcodes per variant. For the GS assay, we were able to get nearly double that. And then finally, we really need to be able to compare between different experimental conditions. This is pretty important for a lot of the applications, and this is something that came up in the questions earlier. So we put a lot of effort into developing a mixed effects negative binomial regression analysis pipeline to be able to actually compare between different experimental conditions. All right, so the reason we want to look at different conditions is because we actually take each library and put them under a variety of different conditions. So again, we developed two different assays for MC4R, the GS and the GQ signaling assays. And we put each of these libraries under a panel of different conditions, either with no stimulation, with alpha MSH stimulation. Remember, this is the native ligand agonist. And we tried multiple concentrations for this. We additionally tried different drugs in combination with our DMS assays. One which is an agonist-like small molecule compound, which I'll talk about later in the talk. We tried multiple concentrations of that. We also tried a chaperone-like drug and different compositional normalization controls. And we run each condition in quadruplicate. So this is a lot of data, and I want to talk through kind of different applications or different questions that we were asking, but this is just kind of a panel of a subset of the different 18 heat maps that we generated for this data. But I want to focus just on like a few core questions that we're really interested in for MC4R and what we can learn from DMS for this program. The first question is, how do variants impact MC4R's different functions? The second is, which patients are actually likely to respond to a treatment? And the third is, can we use DMS to better understand how drug compounds actually interact with MC4R? So first, how do variants impact MC4R's different functions? So this is there's a lot of different ways that we can look at this question because we generated so many different data sets. At a very baseline level, we can look at an individual data set and the heat map for that, or the DMS results for that. So I'm shown here the results for just one assay, the GS assay, at low concentrations of agonist signaling. Showing heat maps, two different versions of the same heat map for the same data, which again, with each variant on the y-axis and the residue position on the x-axis. In our coloring scheme, anything that's dark blue is significant loss of function, and anything that's more in a brown color is gain of function. And I'm showing the top heat map shows the log 2-fold change, and the bottom heat map shows the z statistic or significance of the effect of the variant. These aren't exactly interchangeable metrics, but for the purposes of this talk, since we had really well-balanced libraries and robust statistical support, I'm going to use these somewhat interchangeably in the talk. So, at the baseline, we can look at and see how different variants impact, or how whether or not secondary structural elements are impacted by mutation, and that's shown here at the top is the secondary structure, the alpha helix structures that form those transmembrane domains, and you can see, not surprisingly, that these regions are very intolerant to mutation. We can also intersect this, of course, with this data with clinical genetics data. So this is shown as an intersection with ClinVar data, again, for the same exact assay that I showed on the previous slide. The top variants here are not found in ClinVar, since we obviously tested a lot of variants that aren't present in the human population or aren't currently known to be present in the human population. The x-axis is the DMSZ statistic, so anything that's 0 is wild type, anything to the left is loss of function, anything to the right is gain of function. You can nicely see that nonsense variants typically cause loss of function. Missense variants are more heterogeneous. And then if we look just at the ClinVar classifications, you nicely see that the predicted pathogenic variants are significantly loss of function, more likely to be significantly loss of function than predicted benign variants. All right, and then we're also really interested not just in, pathogenic versus benign, but really understanding are there quantitative impacts of variants? Can we actually see, say partial loss of functions or quantitative effects of variants? And one way that we were looking at this was we found a review paper that had a panel of about 70 different human variants from MC4R and had classified them as to whether they were predicted to be loss of function with mild to no effect, whether they were predicted to have partial effects, or whether they were predicted to have severe effects. And I'm just showing on the y-axis our DMS data for those variants. And I think we're seeing a nice quantitative scale here between the different types of quantitative effects of variants. At another level, we can actually compare and contrast different assays to see if we can identify variant effects that are specific to one assay or another. So I'm showing here data comparing our GS assay versus the GQ assay for a moderate level of agonist stimulation. On the x-axis is the Z statistic for the GS assay. So again, wild type is 0, loss of function is to the left, gain of function is to the right. And on the y-axis, we have the Z statistic for GQ. So anything above is gain of function, anything below is loss of function. So each variant is a different dot, and you can see that all of the variants that are colored in black are significantly loss of function in both assays. Anything that is in gray has no impact in either of the assays. And but what's really interesting are these variants that are in red or purple. So red indicates variants that have a significant loss or gain of function only in the GS assay. And those that are in purple have a significant gain or loss of function only in our GQ assay. So we're seeing some differences between the effects of variants on these different signaling pathways. In particular, we were really interested in looking at these down here where they have a significant loss of function only in the GQ assay. In particular, there's a number of positions where there are at least 2 variants, so 2 different amino acid substitutions that have an impact only in the GQ assay but not in the GS assay. And when we map these onto the protein, these could be pointing us towards regions of the protein that are really important for MC4R to figure out which of the pathways it should bias towards in signaling. All right, and then at a very high level for variant classification, we can look across a variety of our different data sets. And so this is just showing, we asked the question, how many variants have a significant effect in at least one of the conditions under kind of normal agonist stimulation. So this is for both the GS assay, the GQ assay, across all different alpha MSH concentrations. And lo and behold, about 6% of the variants have a significant gain of function effect on MC4 activity, and about half of the variants have a significant loss of function effect in at least one of the conditions that we looked at. So the next question that we were interested in is which patients are actually likely to respond to treatments. We were particularly interested in, since we work predominantly in trying to identify small molecule chaperones to treat protein misfolding diseases, we were particularly interested in looking at the class of MC4R variants that are missense variants that lead to misfolded MC4R, which then lead to it failing to localize to the membrane. There's been one previous, or the idea behind chaperone therapies is that they act just like protein chaperones in the cell, that these are small molecules that will actually go and bind to the protein to stabilize its folding and help fold and therefore lead to it being trafficked properly into the cell membrane. There's been a previous chaperone that's been identified for MC4R, and this is a molecule that's called Ipsin-17. It's a small molecule whose structure is shown here. And so we were interested in understanding which patients are actually likely to respond to this therapeutic. So for this, we did one DMS assay with alpha MSH stimulation alone. Alpha MSH, again, is the native peptide ligand that activates MC4R. And we also did a version of the assay with alpha MSH treated with Ipsin-17, in combination with Ipsin-17. For the sake of clarity, I've narrowed down all of this to a bar plot. So on the y-axis is the GS assay activity. Everything that I'm going to show is going to be relative to this bar right here, which is the wild type protein without treatment. One really nice confirmation that we're seeing something is that in the presence of Ipsin, the wild type actually has increased signal. That's a known effect. The chaperone is known to impact the wild type version of the protein and help it fold and therefore increase its signaling potential. We also wanted a little bit of confirmation that what we were seeing with DMS was real, and so we found a paper that had previously tested a panel of about 10 or 12 different variants with this particular chaperone, and they had classified them as having no rescue, weak rescue, or strong rescue. So really nicely, we confirmed their results for no rescue. These two variants are clearly loss of function under wild type or under no drug conditions, and they have the drug seems to have no impact on their activity. For the weak rescue variants, we don't actually see an effect of the drug in our assay, but for the strong rescue variants, we see a really nice effect of the drug for most, if not all, of the variants that were previously shown to have a really robust response to this drug. So we wanted to expand this a little bit more to other patient variants. Again, we had a list of missense variants from this paper. All told, there were 72 human variants that were reported previously to have a misfolding or mislocalization phenotype. We looked at these to see in our DMS data to see if they respond to chaperone, and a large panel of them do not have any response to the chaperone therapy, but about 20% of the variants in this data set, of these 72 reported human missense variants do actually have a response to chaperone therapy. So these are probably the patients that you want to enroll in your clinical trial for this drug, since they're most likely to actually benefit from this type of therapeutic. All right, and then finally, we wanted to understand, can we use DMS to better understand how drug compounds interact with MC4R? And this is kind of the holy grail for us for DMS, which is to be able to use DMS to actually directly design better therapeutics. And so a big part of drug discovery is medicinal chemistry to understand how a drug actually interacts with a protein. So shown here on the left is a protein, schematic of a protein in green, showing one particular amino acid side chain coming off of that protein, and looking at a particular drug to see if it interacts with that particular residue. The one here on the left does not interact with the residue, but one here on the right shows that you can identify, say, hydrogen bonds between a drug and a residue. And this is really a critical part of drug discovery and development. And so we wanted to know what information about drug-protein interactions can we actually get from DMS? Can we actually really understand how drugs are interacting with a protein? And to look at this, to start looking at this, we wanted to understand, do different variants impact the effects of different agonists of this protein? And so this is MC4R again, and alpha MSH, which is its endogenous peptide agonist. Alpha MSH is the structure is shown here. There have been a number of drug-like small molecule agonists that have been developed for MC4R through various different drug programs. One whose acronym is called THIC, is shown here. It's a small molecule agonist that also binds into the same spot of MC4R to activate its signaling. So we did deep mutational scanning in the presence of each of these different compounds. I'm showing data again just for one low agonist concentration for our GS assay. And you can see that shown here the heat maps for alpha MSH versus thick, and the heat maps are pretty similar. Many variants have exactly the same loss of function effect with either of the agonists. But what are the differences? Which variants actually respond differently to each agonist, and what can they tell us about how the protein is interacting with the drug? And so to orient you to what I'll show you, the plots here are structures looking down into the barrel of alpha MSH, showing where the agonist actually binds to activate signaling. So each of these kind of bubble plots is the alpha MSH. On the left is shown this is MC4R. On the left with the black lines is the alpha MSH bound to MC4R. And on the right in blue is thick bound to MC4R. And each of the different residues in this binding pocket are color-coded. The ones here in yellow are positions where variants disrupt MC4R's response only to alpha MSH. There's a fair number of these residues, which is not surprising since alpha MSH is a fairly large molecule compared to thick. And it interacts with a lot more different residues in the binding pocket of MC4R. What's really interesting, though, is these residues that are colored in green. These are positions where different variants disrupt MC4R's response to alpha MSH or thick, but not to both. And so to kind of explain this in a little bit more detail, I'm going to focus on one particular position, which is residue 129. Which is in green here, back behind the drug binding pocket. So at this position, I'm now showing the data for just this position at the bottom with every possible amino acid substitution and the results of the effect of that variant on alpha MSH or thick signaling, or sorry, alpha MSH or thick stimulation. 0 again here is wild type. So you can see that like putting a stop codon or a D or an F at this position negatively impacts the function of the protein under signaling from either of these agonists. But a lot of the variants have the same effect whether you use alpha MSH or thick. But what's really interesting is that there's a handful of variants that actually have different effects depending upon which agonist you use. So for example, if you have an H, an S, or a T at this position, it negatively impacts the signaling of the protein, but only if it's stimulated with thick as the agonist, whereas there's no effect from alpha MSH stimulation. And if you have a valine at this position, you get the opposite effect, that alpha MSH activation is affected, whereas thick's activation isn't affected at all. So variants at the same position can actually have differential effects on stimulation by different agonists, which is pretty cool. So we think, though, and this is still early days for this particular line of investigation, but we think that in combination with structural information, this would provide a really granular view of protein-ligand interactions that could be better harnessed by medicinal chemists and computational chemists for better drug design. So with that, I'll summarize my talk. We used deep mutational scanning to measure the effects of variants on 2 signaling functions of MC4R. We think a practical application for this in drug discovery is to identify which cellular mechanisms to target with therapeutics. We identified patient variants that respond to chaperone therapies, and so a practical application is to stratify your patients in a clinical trial to identify which ones are much more likely to actually benefit from a particular therapy. And then finally, we identified variants that differentially affect MC4R's response to endogenous and drug-like agonists. And we think that combining these protein structure-function relationships with chemical structural activity relationships could really be used to optimize drug design in a really meaningful way. With that, I will acknowledge a number of people. This work was contributed to by a lot of people at Octant, and both past and present. Particular shout outs to Nathan Abel and Connor Howard, who did a lot of the experiments and analysis for this project. And then shout outs to Jamie Fraser and Debbie Marks for being wonderful scientific advisors on this project and others at Octant. So thank you.
Questions from the floor? Anyone? Again, I'll kick us off. So your assay with the chaperones, where you got different effects, that some responded and some didn't. Mm-hmm. Do you have a molecular explanation for the difference? Is there clustering? Is it stability?
We haven't looked at it in too much detail. There's definitely there's some work in this area too on some compounds that Vertex has developed for the CFTR mutations. And so there is starting to become some literature in this area about like how chaperones probably do affect things that are mutated in similar parts of the three-dimensional space of the protein. Yeah.
Hi. Is it anticipated that if you can drug MC4R effectively for MODY, it would then have applications more broadly?
Presumably, yes.
I think we have a question online.
Yeah, we have online. How would you consider off-target effects of drugs when assayed in a single cell to minimize side effects?
I think it's hard to do that with a DMS assay unless you could maybe do like a multiplex assay for other types of drugs, or sorry, other types of proteins. And so people do these, that they develop these assays where you're testing whether or not like all GPCRs respond to a particular drug. But I think it's not really a DMS assay, but it is another like form of high throughput assays.
Yeah.
Hey, that was great. So in every gene there'll be other variants linked to the disease-causing variant. Mm-hmm. To what extent do you take that into account in your assays? Do you go back through and see whether there's any activity in cis or of other common variation or indeed other variants potentially within the gene?
So you're asking like if there's two different variants in the same haplotype?
Yeah, like a polymorphism, and then you've got the disease-causing variant, and do you explore those kinds of interactions?
We don't really, and again, like we're also kind of limited, and we're not looking at like splicing or anything like that. We're really just looking at kind of one single amino acid at a time at this point. But it's certainly, you could do that in a library context, especially if you were like wanting to look at like say suppressors of a particular variant. Yeah.
I think we just have one more question.
So I wanted to ask about using the z-scores,
Mm-hmm.
because these are non-normal and many times multimodal distributions. So what is it's okay to use them, and what's the interpretation?
This might be a better question for me to put you in contact with our bioinformatics team. I think in large part, especially for this particular set of assays, we do see, like they recapitulate the fold change pretty well. That's not the case for all of our assays. So I think that there are times where we can use them somewhat interchangeably and times that we can't. But maybe we can take this question offline.
Great, thank you very much. We'll move on.
Thanks.